All right. People are still dropping in, but I think we'll start today with Fluxim's 19th webinar on the topic of OLED degradation. We will have first the contributions of our two speakers, and then after the two talks will be question and discussion round. So please post your questions into the chat during the talks, and we will go through them after. So it's my pleasure to introduce Chris Giebink, professor at the University of Michigan and the head of the Applied Optoelectronics and Photonics Lab. Chris' research focuses on the fundamental physics of organic semiconductors, specifically photonics, including charge transport, excited states, light-matter interactions. His work is highly relevant to OLED, solar energy, and a broad range of optoelectronics devices. So we are very pleased to welcome you, Chris, today to share your insight into mechanisms and modeling of blue OLED degradation. Thank you very much for joining, Chris. You can share the screen, and the floor is yours. We can see the slides.
All right, thanks, Matthias. So as you said, I'm going to talk a little bit about some of our recent work trying to understand why blue OLEDs degrade and how we model them. Just a quick overview. I'll talk about a mechanism that we call the P2 polaron pathway for blues that we think helps explain why blue OLEDs in particular are more unstable than greens and reds. And then we'll talk a little bit about our efforts to model OLED degradation by building it into drift-diffusion based OLED models, and then bringing in kind of a second look at the way that trap-mediated recombination happens in organic semiconductors, particularly when excitons are involved. So let's start off at the beginning. When we think about OLED degradation, we usually divide it into two basic classes. The first is extrinsic degradation, and this is degradation due to things like fabrication and packaging. These are the things that you can control. Good examples are moisture-induced dark spot formation and particle-induced micro shorts and catastrophic failure. On the other side of the coin, there is intrinsic degradation, and this essentially corresponds to breaking apart of molecules due to the processes that normally happen in OLED operation, so running current through and emitting light. And the hallmarks of intrinsic degradation are luminance fade over time, so the luminance fades at constant current density, and then the voltage rises at constant current density over time. Now, the picture that we have for intrinsic degradation usually looks something like this. So we've got a generic OLED, the whole transport layer on the left, electron transport layer on the right, an emissive layer here in the middle. And usually your charge carriers and your excitons are concentrated in some portion of the emissive layer. We call that the recombination zone. And then for various different reasons, molecules can break. So you're essentially breaking a bond, and then that leads to molecular fragments that may undergo secondary reactions. When all is said and done, we call that a degradation product. And degradation products can be bad from a light emission standpoint for a couple of reasons. The first is that if they have energy levels within the transport gap of your organic semiconductor, they can act as traps and lead to trap-mediated recombination, which is non-radiative usually. They can also have excited states potentially that can accept the energy of already formed excitons, and so that leads to luminescence quenching. Both of these lead to loss of light and luminance fade over time. Now, the question has always been, what are the processes that lead to the formation of these degradation products? And there have been a whole bunch proposed. I'm going to focus on four that we think are especially important. The first one is that charged molecules are unstable. We always have charged molecules in OLEDs because we're conducting electrons in holes. So we've always got cations and anions. And there's good evidence that in certain cases, things like the ALQ cation, for example, are known to be unstable. Second mechanism is just that excited states, or your excitons, are unstable. You potentially have a dissociative pathway crossing one of your excited states and end up kind of going from your singular or triplet excited state onto this dissociative pathway. Good evidence for this as well. And then these last two involve things like annihilation. So you can have upper excited states of molecules that are unstable. Sometimes those get populated through exciton annihilation reactions, where one excited state donates its energy to another one. And then you can also have energy that's donated to your cations and the anions in the device. And so in this case, you can have excited cations and anions that are also unstable. Okay, so when we think about these different mechanisms, thinking about the blue problem actually puts them in sort of a useful context. The blue problem has to do with the fact that blue OLEDs in general are much shorter lived than their green and red counterparts, often by more than an order of magnitude. It's been a big problem for industry over the years, and it was kind of nicely summarized in this paper here, just looking at the lifetimes of a bunch of different color OLEDs as a function of their emission wavelength. And at first glance, it kind of looks like a scatter plot, but if you look at the Pareto frontier, what the general trend is that your OLEDs live shorter if they emit shorter wavelength. And so if you look within a given class of emitter, fluorescent or phosphorescent, you generally have this sort of edge here that drives this correlation with shorter lifetime and shorter emission wavelength. So the question for us for the past couple of years has been, why are blue OLEDs in particular uniquely unstable? And so the observations that we made in the beginning went like this. So the first is that if you trade out your blue emitter for a green emitter in otherwise the same architecture, you can generally improve your device lifetime. Lifetime generally gets worse the bluer you go. We just saw that in the previous view graph. And then fluorescent devices in general can be made to live longer than phosphorescent or TADF devices. And if we just look at these three observations, the conclusion would be make a more stable emitter, which there is certainly a big piece of the problem that involves just making a more stable emitter. But then there's some other things that don't quite fit so neatly into just the emitter being the problem, because anecdotally, what you find is that the degradation of other molecules in the device, things like HTL, ETL, host molecules, also tend to speed up in bluer devices. And that's not so obviously connected to the stability of the emitter. And so if we go back and look at some of these degradation mechanisms, this notion that excitons are unstable, lots of good examples of the emitters being unstable. You see that just in the photodegradation of blue phosphors like Fur6 and Furpic. Even some of the hosts like CBP are known to be unstable. And then some of these annihilation-based pathways, in particular exciton polaron annihilation, which is something that we've long thought has been important. You see good evidence for that as well, and the experiments to that effect tend to look like this, where you have a device and you excite it optically, you run current through it, and then if you excite it optically while running current through it, you see a synergistic effect and much greater degradation. And so I want to focus on this possibility, and it's a little bit unsatisfactory because you can draw diagrams like this. It's not very useful because it doesn't tell you what the specific annihilation reaction is that's causing the problem, really what states on what molecules are involved, and then why exactly is it worse for blue devices. It's easy to sort of wave your hand and say, well, there's more energy flying around, therefore things should degrade faster. But again, that's not being very specific. And so the last thing then is, of course, why would any of this cause molecules outside the recombination zone to degrade faster as well? So I want to start by going back to some of the basics of excited states. First is just the exciton transition that's dominated by the HOMO-LUMO transition that we're all familiar with. And then look at the optical transitions of charged molecules. So for example, if we take one of the electrons out of the HOMO, we now have this partially vacant HOMO, or really a SOMO, and that opens up two new transitions, one from lower lying molecular orbitals up into that SOMO now, and then one of the remaining electrons up into the LUMO. We call the lower one the P1 transition, and the upper one the P2 transition. And then if you were to doubly oxidize the molecule, you now have this dication transition, this single transition here. And a good example of these is this classic whole transport molecule, TPD. It is normally transparent in solution. That would be this side over here, because it only absorbs in the UV. That's the black line that I'm tracing. And then if you oxidize it at its first oxidation potential, your solution turns orange, and that's because you get the appearance of two new cation transitions. One in the near infrared out here, that's the P1 transition, and then one in the blue over here, which is what we call the P2 transition. And then if you oxidize it at its second oxidation wave, you get a green solution, and that's because of the emergence of the dication transition here in the red. All of these transitions are evident in thin film. So this is now thin film of TPD doped, P-doped with 10% moly oxide. You can see P2, P1, as well as what's now the bipolaron transition. Now, another molecule is also another whole transport molecule, TAPC. Has all of the same features. You've got your P1 transition in the red, P2 transition in the blue. And one of the interesting things about TAPC is that its cation has measurable fluorescence. So there's actually a Frank Condon mirror of these P1 transitions here that will fluoresce, albeit very weakly. But you can measure it. And one thing we noticed early on was that if you excite that fluorescence via either the P1 transition versus the P2 transition, you see very different signatures in terms of stability. So when you excite it with P1, the fluorescence is stable over time. But as soon as you start exciting with the P2 transition, that fluorescence degrades very rapidly. And so it made us start to wonder whether this P2 transition is a pathway to instability for the charged TAPC molecule, and whether that might be more general in blue OLED materials more broadly. And so since then, we've looked at a range of different molecules, host molecules. These are all carbazole-based host molecules, whole transport materials, electron transport materials, and various different dopants, phosphors, fluorescent molecules, TADFs, things like that. And because I'm kind of limited on time, I'm just going to focus on the carbazole-based hosts as representative of the results for pretty much everything that we've looked at so far. And so if we look in particular at CBP, or actually it's a tert-butyl variety of CBP, you can chemically oxidize it in solution. And again, you get this P1 transition in the red, this P2 transition in the blue. And you can track the degradation of those cations just by looking at the absorption band. So this is now exciting that solution of cations at say 405 nanometers. So we're exciting P2, and you can see that the absorption bands of the cation steadily degrade over time. And so if we track the loss of the cation over time and we compare that with the loss of, say, a neutral molecule, a neutral solution excited much more deeply in the UV, so this would be related to just photodegradation of neutral CBP, you can see that the P2 excited cation degrades much, much more quickly. You can go the other direction, which is something that our colleagues at USC did in Mark Thompson's group, and there they reduced MCBP with potassium in solution. And again, you have very similar P1 and P2 type transitions for the anion now. And they find that when they excite it, the P2 in the blue, it leads to rapid degradation of the anion, whereas they don't see any degradation on this time scale anyway, if they excite the P1 transition. And so this, again, seems to suggest that these P2 excited cations and anions are much less stable than your neutral excited molecules. You can go a little bit further. So if we go back to the cation now, and we systematically vary the wavelength with which we're degrading. So we go from the near infrared all the way into the UV, and it's a monomolecular degradation, so we can fit it just with a first order exponential. And that allows us to extract a degradation rate constant for what is nominally the same photon flux for all these different wavelengths. And then if we plot that degradation rate constant as a function of the degradation wavelength, we get these blue dots here. And so this is sort of an action spectrum for the degradation of the cation. And what you can see is that it seems to trend up right around where that P2 transition happens. Exciting P1 is not problematic, exciting P2 is. And so this is sort of further reinforces this notion that P2 is unstable. And at this point, I'm going to have to switch gears here, but I'll just say that we've made similar observations in thin film for a wide range of whole transport, electron transport, emissive layer, host and dopant molecules. This appears to be a general degradation mechanism. We see very specific degradation products come out of these reactions. And so it is kind of revised our picture of why blue OLEDs in particular are unstable. We look at all of the different molecules that we use in a blue OLED. They're all going to have P2 transitions in the blue, just by virtue of having their neutral excited states in the UV. And that means that when we go from a green emitting OLED to a blue emitting OLED, our blue spectrum starts to increasingly overlap with these P2 absorption bands, and therefore increasingly excite these unstable cations and anions in these various different layers of the device. Now that excitation can happen via a couple different mechanisms. If it happens via the near field, this is mostly via Forster process. We've got a strongly allowed absorber. We've got a very efficient emitter, so Forster will be efficient. But of course, just because we have a lot of blue light flying around inside this device and we have strong absorbers, you can have far field transfer, or just reabsorption of blue light by these anions and cations. So this provides maybe one additional explanation for why blue OLEDs degrade faster, because you're increasing the spectral overlap with the P2 anion-cation transitions. It also explains why molecules elsewhere in the device that are ostensibly far from all the action also degrade faster in blue OLEDs. And it explains why stable dopant emitters don't fix the problem, because even if you've got a rock-solid emitter, you haven't fixed the instability issue for all the rest of the molecules in your device. Okay, so I will switch gears now and talk a little bit about how we've used this information to try and model OLED degradation. OLED degradation modeling is kind of divided into two ends of the spectrum. On the simple end and the very sort of coarse end of modeling is a rate equation-based approach. And this is sort of a pen and paper approach that neglects all the spatial aspects of your OLED degradation. And then on the other end, you've got very sophisticated Monte Carlo models that model things in 3 dimensions at the molecular scale and can tell you exactly where things are degrading. Both of these have lots of parameters, and therefore, maybe unsurprisingly, can do a reasonable job fitting trends like luminance fade in both cases. Something that we've looked for is maybe a compromise between these two that brings into account the spatial variation in the degradation, but is maybe not so computationally demanding as a Monte Carlo model. And so that has led us to try and incorporate OLED degradation kinetics into drift diffusion models like Setfos. And so as we were doing that, one of the things we realized is that we didn't have a good idea really about how mid-gap levels lead to trap-mediated recombination. In inorganics, we call this Shockley-Read-Hall recombination, but there's always been a little bit unsatisfying trying to port that over to organics because when we look at this process, let's say we have a discrete level inside the transport gap of an organic semiconductor. Recombination is sort of a balance between electron capture processes. You can thermally re-emit your electron. Same thing, you can capture your hole and thermally re-emit your hole. And if you go through the detailed balance of this, what you come out with is the usual Shockley-Read-Hall recombination expression. But the problem is, one of the problems has been, what are these capture coefficients? So these are always sort of empirical coefficients, but we really haven't had a good idea of what they are in organics. In inorganics, they're parameterized in terms of things like thermal velocities and capture cross sections. Both of those are inapplicable concepts for organics, where things are diffusive. And so we started to think about this in the context of exciton annihilation, where capture coefficients are well known for a diffusive process. You define it in terms of your diffusion coefficient, say for an electron, if we're looking at this electron capture process, and then the capture radius, which in this case corresponds to the radius of a molecule. So those are both pretty well known. That describes this initial electron capture. But then as soon as that trap level is now charged with an electron, at this point, the recombination process is Langevin, and the capture cross-section at that point is just a Langevin coefficient given the mobile hole over here. And so if we incorporate these capture cross-sections, then now we've got a Shockley-Read-Hall expression that is defined in terms of well-known stuff like electron diffusion coefficient and hole Langevin coefficient, just given in terms of the hole mobility. But then there's another complication when you're dealing with organics, because in many cases, you're dealing with closed shell molecules and the possibility of forming excitons. So in this case, let's imagine a scenario very similar to what you have with an emissive dopant inside a wide gap host, like you often have in OLEDs. In this case, you can trap your electron in the LUMO, detrap it. You can trap a hole in the HOMO, detrap it. If you trap an electron and a hole at the same time, you form an exciton, which has a finite lifetime, and also a finite probability of redissociating. And so if you take all of these processes now in a detailed balance, you also get an analytical result. It's much longer than the usual Shockley-Read-Hall expression, but it simplifies for certain simplified scenarios. And so one of them is if your energy levels of your guest are straddled symmetrically within your host, and if you set the electron and hole concentrations equal to each other. And in that case, the numerator looks exactly like your Shockley-Read-Hall expression, but your denominator has a little bit more. So the term on the left over here corresponds to standard Shockley-Read-Hall recombination through the parallel combination of the LUMO and the HOMO. So this is just treating the LUMO and the HOMO as two independent trap levels. But then there's this additional contribution in the denominator that depends on things like exciton binding energy on the guest, as well as the CT binding energy of the guest host system. And in general, this term slows things down. And so the impact of including excitons in Shockley-Read-Hall recombination is that it slows down the recombination rate. You can see that here. So what I'm showing is the normal Shockley-Read-Hall recombination rate of the HOMO and LUMO in parallel. That's the black line. And then when you include the exciton physics in this, that's these orange circles here. And you can see they're exactly the same for the case of a nested system, but they start to differ as soon as you start to offset and stagger your guest and host energy levels. And in particular, when you're significantly staggered, something like this, where you've got a type 2 offset, your excitonic Shockley-Read-Hall recombination rate goes down by orders of magnitude. And physically, this is easy to understand, just because we've now got a type 2 heterojunction. So as soon as we form the exciton on the guest, it has an increased probability of redissociating back into free charge carriers on the host. Okay, so now that we can describe trap-mediated recombination, we incorporate that into an OLED degradation model. Again, we're going to keep this simple, 3-layer device. Everything's happening in the emissive layer. We're going to take one particular description of defect formation. We're going to assume that it's proportional to exciton hole annihilation. So this would be excited cations, or what's leading to degradation. And for each excited cation that we create, it has a degradation probability, albeit very low, that it leads to a defect that leads to a mid-gap state wherever it's formed. And so we can plug this into Setfos and also compare it with a pen and paper rate equation model just to see how close the two are. And what we find is that both predict similar functional dependencies in terms of the luminance fade over time. Here's the rate equation in blue, and then the Setfos using exactly the same parameters is in red here. And so we're predicting similar functional form of degradation, but the fact that we're not accounting for the spatial variation of degradation in the rate equation model leads us to get the lifetime wrong by about a factor of 3. One other thing that I'll point out is that it's important to do this degradation simulation iteratively. So what you need to do is, because defects will change the electrical characteristics of your device, which in turn changes the degradation rate, you need to simulate the charge carrier and exciton distributions in your device, run some of the degradation kinetics, and then re-simulate the charge and exciton distributions and do that in a feedback loop. Otherwise, you get very different results. So the iterative approach is here in red, and a non-iterative approach is here in the dashed line. Nonetheless, you can sort of fudge the parameters in the rate equation model and get them pretty close to the Setfos model. That's the dashed blue line here. So that kind of explains why rate equation models can model things reasonably well in terms of what you see in experiment. One of the surprises in this, though, was in the voltage rise that this predicted. So in this case, the rate equation model predicted a few tenths of a volt voltage rise, very similar to what you would see in experiment. Drift diffusion model, on the other hand, predicted almost no voltage rise, and this was a little bit of a surprise for us. And so we looked at it in a little bit more detail, and the reason that it happens is, let's say that you take a hole-only device and you introduce some traps right here in the middle. Normally, those traps would be fully occupied under forward bias, and so you end up with some fixed charge that will lead to the voltage increase. And just running through sort of a Poisson equation calculation, you predict a voltage increase that looks something like this, and that's in very good agreement with what you get for a drift diffusion model. But as soon as you put those traps near a heterojunction, where you're already accumulating a high density of free holes, all of a sudden your Poisson equation, just assuming that the fixed charge from these trap states, that voltage increase no longer agrees with what you actually get from your drift diffusion simulation. And the reason is because when you have a very high density of free holes to start with, and all of a sudden you put in this fixed charge of occupied traps, the free holes will redistribute in order to minimize the voltage increase. And that's even more the case if you go from a unipolar hole-only device to a bipolar device, like an OLED. And so what this means for us, when we go back and say, assume that these traps are forming just in the emissive layer versus, say, in the hole transport layer or in the electron transport layer, is that in regions like in the emissive layer recombination zone where the free charge density is high, you see a very different voltage rise than if your traps are forming elsewhere in the device. And so what it suggests to us is that luminance loss and voltage rise don't necessarily originate from the same set of defects. Voltage rise seems to be most significant when your defects are forming outside regions with high free carrier density, say, for example, in the HTL or DTL, whereas your luminance fade is dominated by degradation products that are formed in the recombination zone in the emissive layer. Okay, so I'm out of time. I will just summarize quickly. I told you about this P2 degradation pathway for blue OLEDs. The excited cation and anion P2 transitions are unstable, and increasing overlap of blue emission with those transitions leads to faster degradation in blue OLEDs. Talked about this organic version of Shockley-Read-Hall recombination and how it's formulated for organics in terms of diffusive trapping and Langevin recombination. And then lastly, about trying to incorporate degradation kinetics into drift diffusion models. I think Setfos actually has a module that does this now. And so with that, I will thank the students that did this work. Majority was done by Dr. Adrian Pisano, Nina in Mark Thompson's group at USC, Mark and Peter at USC, Steve Forrest, colleague at Michigan, Sukrit Mukherjee, formerly at Dow, and then NSF, DOE, and Merck for funding this work in various different stages, and you for your attention. So thank you.
Thank you very much, Chris. Your time is truly appreciated. Thanks for the nice introduction also into the blue OLED degradation. And at this point, I mean, yes, you're right. Like it was recently released in Setfos 6.0 degradation model, and I like to mention at this point, if I'm not wrong, that it was you and your PhD, Adrian Pizzano, who contacted us several years ago and kicked off this whole degradation discussion within Fluxim. So as I said, questions, you can continue to ask questions. I will ask them in the end then together with Vasilis' part. So then we continue at this point now with our second speaker, Vasilis Georgakopoulos. He's a PhD candidate in computational physics at the University of Bayreuth and my dear Fluxim colleague. Vasilis works on OLED technology with a focus on electrical and optical characterization and multiphysics simulation as well. And today he will present the work that connects advanced polymer LED modeling with practical understanding of degradation processes in devices. Thanks, Vasilis, for joining. You can share your screen.
Thank you. Thank you for
and then
having me. Let me share my screen. So
yeah, all good.
All good? Okay. So as Matthias said, yeah, today I will be talking about temperature-dependent electron trapping in the semiconducting polymers, and specifically in a very well-known and very well-studied super yellow polymer, PPD copolymer. So the motivation for this study comes from earlier work that it has been identified that for polymers, there is this phenomenon where water and oxygen impurities penetrate the polymer matrix. And in the presence of electron charge, a reaction can be catalyzed where there is a complex of two water molecules, an oxygen molecule, and an electron can form a stable complex that can act as an electron trap. And this complex has been identified to sit around more or less 0.6 or 0.7 electrovolts below the LUMO level, and it acts as an electron trap. So experiments were performed to see how exactly the dynamics work in EMPA, a study conducted by Sergi and al. In Switzerland. And they used basically the temperature dependence. They fabricated electron-only devices with super yellow as the active material, and they used the temperature dependence to identify the dynamics. Because the theory was if this complex water-oxygen complex is responsible for the degradation, and then the diffusivity of this polymer should play into the dynamics of trap formation. So by increasing the temperature, they could show that they increase the diffusivity, so more trap for more precursor molecules meet up, and they form more trap sites. And when they did this experiment, we can see in the left graph that basically the current decay follows this trend, where as we increase the temperature, the current decay increases for the same amount of time. And we can also perform electron trapping simulations with Setfos and simulate the current decay. And we can see that we can extract with this way, we can extract the trap density basically inside our devices. And we see that also the trap site density increases with a higher temperature. So this process is catalyzed basically by temperature. It's a diffusive process. And the dynamics are as expected. So then we moved on to trying this with PLEDs, with the bipolar devices, because this is the interesting application basically. And what they found out, the peculiar thing was that basically at room temperature, when they did the same thing for a PLED device, while for the electron-only device, they saw degradation in the current of about 80%. For the PLED device, it was only about 10% or so. And this was a quite strange phenomenon. So why is it so little, the degradation compared to the electron-only devices? So one characteristic thing of these traps is that they are metastable. So basically, when we remove the high electron density from inside the devices, these trap sites basically disintegrate slowly. But if you wait long enough, and then you can perform another run at the device, and you can see that the current performance has been restored. And they did these experiments for the PLEDs also, and they saw that the current basically recovers. If you wait long enough, it recovers to its initial value. So they are indeed electron traps formed by this water-oxygen complex. And so why is the impact on the device performance so different than the electron-only device? So then they moved further to we conducted together experiments. We took different data sets, multiple devices, different experimental setups, and all the times what we after statistics of course found out, but that the temperature dependence was also different from the electron-only devices. So what we saw in the PLEDs is instead of a monotonic increase in the current decay, we saw that there is this U shape. So basically, the current decay, of course, is much smaller in magnitude, about from 10 to 20%. And the same was also was observed for the emission. So what is happening? Why is it so different? Why the electron traps affect the PLED in such a different way? And to discover this, we performed some simulations. But to do degradation simulations for an OLED, we first need to start from the pristine device. So we need to have, it is essential to have a very good model, well-calibrated model for the pristine device if we want the results for degradation to be relevant. So for this purpose, we took JV curves for the pristine device and also measured the luminance for different temperatures and included some of the current data that we have for the unipolar and electron-only devices. And we chose to model the mobility was basically usually the most important parameter, when the rest of the parameters such as injection and energy levels of the active material is known. The most important parameter, which is usually influenced by fabrication conditions and stuff like this, is the mobility. And to calibrate this, we use this data, we perform simulations, and we used a mobility formula, which has a field and temperature activation factor that is usually applied to model the mobility of organic materials like Super Yellow. And what we found out basically was that there was a higher activation energy for the holes and the lower activation energy for the electrons. So the holes were more temperature activated than the electrons. So we would expect that moving from a lower to a higher temperature, the charge balance inside the device would be affected. And then moving to the trapping simulation, we have our calibrated model, the pristine device. So what we do is we now try to include some traps. And the trap level, as I said before, is well known. And for the distribution, we assumed a Gaussian distribution in terms of energy with a fixed energy width. And for the capture, and the other parameter that usually influences a lot the results of a simulation, as the previous speaker also talked about, is Shockley-Read-Hall recombination. And the big unknown also in our case, which we struggled a bit, or like we had trouble finding out how is it exactly should be modeled, was the capture coefficients. And for the holes, the capture coefficients is straightforward because we have a trapped electron and then we have a free hole, and this mimics basically a recombination event. So we can use the Langevin formula to model this effect. But then for the trap that is formed, metastable trap that is formed and dissolves and then it needs to capture an electron. A lot of things go into this process, and it is not exactly clear which, if there is a theoretical formula, how you should approach it. So we varied this value. We used a constant value, different constant values, and we varied it as a kind of free parameter to see what can explain our results. So for the degradation fit, as I said before, what we do is basically we stress the device at constant voltage, and we measure the decay of the emission and the current. And for the simulation, it is basically what we did here is kind of a pseudo-transient simulation where we treat as each point in time as a separate state of the device, kind of. And we start with the calibrated pristine device model, and then we increase the trap density until we can reproduce the emission data for a specific point in time. Then we repeat this for all points in time, and we assemble the transit, as we can see, for example, for the intensity in figure A. Then we can also plot the results for the current in figure B for the same trap density. Since we can plot the corresponding results for the current for the same trap side densities. And what we saw is that basically we can always fit the emission data because this is the target of the optimization. But then for the current, we have some divergence, especially in higher temperatures. And one of the reasons for this is basically we always consider the uniform distribution in terms of traps, but we can imagine, someone can imagine that the distribution or the formation of trap sites will not be uniform once molecules start to form traps, and the availability of molecules in certain places becomes more rare. So we also used a distributed trap model, and we binded the probability of trap formation to the electron trap density, which improved kind of our results, but not they were still not perfect, so a bit of work, maybe still a more realistic, physically realistic model may be needed for such a description in this particular case. And then what we did is basically we looked at the results. So what is the extracted density that we get out of this model? I mean, we were more or less for different electron capture coefficients, we were able to get a decent fit most of the times. And but what we got actually out of it as a fitted trap distribution was surprisingly different. So the electron capture coefficient is a very important parameter if you want to list the trap density as a final result and be able to compare across different studies. So what we saw is that when we included a very large capture coefficient for the electrons, we got a really low trap density, trap site density, and kind of a temperature independent results, as you can see in figure. So basically, in figure A, number 1, we can see that basically all the trap density versus time is kind of temperature independent. They all arrive at basically the same value, more or less. And then we slowly increased the capture coefficient until about 5 × 10⁻14th. And we saw that then what we actually got when we chose a way slower capture rate, we got this, as expected, increased trap density with temperature, as we would expect from the fundamental mechanisms, because we would argue that the fundamental mechanism in the PLED for trap formation and in an electron-only device stays more or less the same. And we expect to find basically the same trap density that we find in the unipolar device, more or less, or at least the same order of magnitude, but not 1 order of magnitude smaller. So by choosing this value for the capture coefficient, the trap generation becomes temperature dependent, as expected. And we also saw that at high temperatures, traps become basically they change. The occupation level changes because at smaller temperatures, 260 Kelvin, we have a high occupation of around 0.4, so about half the traps are occupied. But then when we move to higher temperatures, they get depopulated. So we generate more trap sites, but less of them is actually occupied and active. And we will see, in order to understand this phenomenon, we need to look at the spatial variation of the density profiles. And for a fresh device, we see that basically what we have is we have the holes being strongly temperature activated. So we see that in the bulk of the device, at low temperatures, 260 Kelvin, the holes they don't have such a strong presence there. So at the part where there is a high electron concentration, we'd expect that the traps is going to be filled, and the electron traps is going to be filled, and there is not going to be a lot of SRH recombination. But then as we increase the temperature, we see that the holes start to proliferate across the device. The temperature activated transport kicks in, and they slowly start to have a decent presence in areas where they are, we call them electron rich, or the trap occupation is high. And this can very well be seen if we look at the trap occupation function. And we can see that, as we said before, at low temperatures, 260 Kelvin, there is occupied traps inside the bulk, but at higher temperatures, the traps that is occupied is basically relegated to a small area close to the electron injecting electrode. And this is because of SRH recombination. So there is free holes that recombine with trap electrons, emptying the traps. And we can also plot cumulative plots to understand this. And we see that basically we can see this. We can see the SRH recombination that at higher temperatures, 15 Kelvin, starts to pick up. And this also causes a deviation between the degradation of the current and degradation of the emission intensity, because the total current is given as the sum of the SRH current and the Langevin current, but only the Langevin current leads to a photon emission. So as the SRH current increases, then for the same amount of current, we have less photon emission that recombination that results to photon emission. And we can see this when we reach high electron trap site densities. We see this divergence in the higher temperatures, whereas in the lower temperatures, the Langevin current and the total current stay more or less on the same level. So finally, as a conclusion, what have we learned? In PLEDs, the fundamental process for electron trap formation can be the same as in electron-only devices. The only thing that we have to do is we have to make some assumptions. And it's assumed that the temperature dependence and the final electron trap density should be same as the electron-only device. So basically, we can assume that the fundamental process doesn't change, and then we can, by adjusting this capture coefficient, basically generate meaningful results for the PLED also. Also, the PLED temperature-dependent current displays this U-shape in terms of degradation. It can be simulated if we consider the electron hole asymmetry in the mobility temperature activation and allow constant value for the electron capture coefficient. And third, it is the holes that reduce the impact of individual electron trap sites at higher temperature by emptying the electron traps through SRH recombination. So the takeaway message is that the picture, what is the picture inside the PLED at low and high temperatures? So at low temperatures, the traps are about 40% filled in the PLED. The traps are still present inside the bulk, and the current efficiency is more or less constant at the final trap density. Whereas at high temperatures, the traps are more or less than 10% filled. The traps are confined. The trap sites are confined near the aluminum, or rather, the occupied traps are confined near the aluminum electrode, where we have high electron density, and the current efficiency drops at higher trap densities. So that is it, basically. That concludes the presentation. Thanks everybody for your attention, and I believe now there is a comments section, right?
Thank you very much, Vasilis. Quite nice and quite a complex modeling study you did there with temperature variation. So yeah, let's kick off a question session. I will start with some questions for Chris. Meanwhile, then people might ask some more questions for you, Vasilis, in the chat. Some of the questions I will read directly, but Chris, I start first with a rather broad question. I'm personally interested, like in your experience, in your modeling experience, quite a extensive one as well. What would you say, what types of degradation related questions can simulation answer particularly well, and where would you say that experiments still remain essential?
I think experiments are still essential for uncovering the underlying photophysical mechanisms of what's causing degradation. But as soon as you understand those, then you can plug that into the simulation. And that starts to be able to do two things, I think. One, start to identify relationships. Like, for example, for a given amount of luminance fade, is there always the same amount of voltage rise, for example? The answer in general is no. But understanding how those two observables relate to one another is something I think you can get out of simulation, in particular drift diffusion, OLED degradation simulation. And then the other thing that simulation, of course, allows you to do is start to optimize things. If you know what's causing the underlying degradation processes, you can feed that into how you design your OLED. You know, we already optimize our OLED for high efficiency and spectral purity and viewing angle dependence. Why should we not be optimizing it for longer lifetime? And that's something we've never really had the capability to do without a simulation that is rigorous enough, like drift diffusion, to guide us.
Thanks, yeah. So let me ask the question Kushdeep Kaur was posting. Hello, thank you very much for the very informative talk. Can degradation kinetics of blue emitters be predicted by their thermal properties like glass transition temperatures or by their relative charge mobilities?
Let's see. So there's certainly morphological things like glass transition temperature, properties like that are important for OLED degradation. I think you can at least screen for very weak materials. If you have a material that's going to glass at 60 degrees Celsius, for example, you know that that's going to be a non-starter. So you can screen weak molecules, but once molecules pass that screening, they have a sufficient glass transition temperature. Then you kind of have to go to the next level of screening, which is their photodegradation stability. Make sure that the excited state itself is stable, and then I would, based on what we've seen, start looking at the stability of the excited anion and the cation. So you're kind of taking the lowest hanging fruit first, making sure that things aren't going to morphologically change on you, then making sure that the excited states are stable, then making sure that the anions and the cations are stable, then the anion and cation excited states are stable. So there's sort of this hierarchy that you're looking to do in order to qualify materials for stability in OLED. I don't think the mobility of a given material is necessarily a great correlator with things like stability.
All right, thanks for your answer. Then Dinesh asked, since excitonic Shockley-Read-Hall type recombination primarily contributes to emission in organic semiconductors, can the homo-LUMO offset between the host and dopant create trap states such that the host to dopant transfer occurs primarily through charge trapping rather than purely through thread?
I think in many cases, a lot of the post-gas systems that we use in OLEDs, which have either the HOMO or the LUMO serving as a strong electron or a deep electron or deep hole trap. In all of those cases, a large fraction of the emission is direct charge trap formation. So direct trapping of, say, the electron on the electron trap, and then a hole coming along and recombining with it. So it's direct exciton formation on the guest, as opposed to exciton formation on the host and then fret to the guest. It's easy to kind of go through the numerics of that and see that your trap-mediated recombination tends to dominate in those types of scenarios. In situations where you have shallow trapping, it's probably not quite so lopsided. There, fret probably plays a bigger role.
He also had a follow-up question, which is a bit long, but I split it up. But basically, he asks about, or he thinks that the expression you have for Shockley-Read-Hall, excitonic Shockley-Read-Hall recombination, considers that electron and hole density is equal. Is this true?
Only in that simplified expression that I showed. So the full expression that is general, that can have any electron and any hole density and any set of trap level energetics, that analytical expression is about a page long. It only simplifies down to the short one that I showed you, just for the very simplified case of symmetric energy levels with equal electron and hole concentrations. But the full expression is more general.
Okay, so basically he asks then long about how would you deal with this, but then you also use for if you do full drift diffusion simulation, you anyway consider the actual hole and electron density. Okay, correct. Then I think the question should be answered like this. What I wondered regarding Vasilis, regarding the modeling, you mentioned you used a pseudo-transient. Would the transient degradation model that is now included in Setfos help you in any way, or was this pseudo-transient version sufficient in your case, in your work?
Well, the main thing is that the previous work proved that the dynamics, the kinetics of this trap formation rate is third order, and in the current model there exists, I think, a constant coefficient which doesn't necessarily cover that. So I think the idea is that we are more flexible in terms of the kinetics, and that we can simulate in this way. It is more difficult to perform the simulation, and you have to do an individual simulation for every point in time, but you have more flexibility in terms of the kinetics that you can simulate like this.
Yeah. And also related to the simulation you showed, I was wondering, like, what would you say how relevant it is to understand the distribution of trap formation within the device? Like, both of you talked about this, also Chris, but maybe in Vasilis, in your view, would you say it's essential to look at, well, how traps evolved within the device?
I would say that maybe it's becoming more important. We saw some discrepancy between the emission and the current, and the simulated current and the emission, but certainly would help if we knew exactly how does the kinetics work, as basically reactance is consumed inside the device. So the assumption that the probability, I would guess that eventually, as oxygen and water molecules are being binded into electrode traps, the availability of such molecules in a specific locus inside the device would kind of affect the formation probability. And this is another level of complexity that you could add to the model that we didn't consider in this study. But yeah, there is always more complicated models. The question is whether they should be included, whether they can explain different discrepancies. But
yeah.
We haven't. We didn't go this far.
So Chris, I think if I'm not wrong, you showed some slide where you mentioned that it is relevant. You feel it's relevant to look into profiles. So is this would you say that's a general? You would agree?
Yes, absolutely. I think it's critical. It is something that we didn't quite appreciate in the beginning. We had always assumed that luminance loss and voltage rise were coming from the same set of traps. Seems pretty clear that that's not the case. And depending on where the traps form in your device, it strongly impacts the impact on the voltage. You can have the same concentration of traps, same energetics in one spot and have a very small effect on the voltage, and you move it to a neighboring layer 50 nanometers away, and it has suddenly a volt change. So being able to simulate and understand that is really critical, in my opinion.
Depending on how this is implemented, this adds a huge dimension in kind of parameters. Depending on how it's implemented, like, do you feel? Would you say?
This has always been the problem and challenge in OLED degradation modeling, and in general, is just that the parameter space explodes, and it's very difficult to constrain it in a unique way. I think that's one of the challenges going forward is how do you constrain it to a degree that it's making meaningful predictions rather than just fudging around with the parameters. So I don't have a great answer for that. But I think as we start to understand more directly what's driving the degradation of the particular molecules that we're using, we can start to kind of narrow things down a little bit and constrain things better. But it is an ongoing question for sure.
All right, let me see. That's actually rounds up the whole degradation discussion quite well, this answer. I also don't see another question in the chat, I feel like. So I would say we wrap up the webinar. Thank you very much, both of you, Vasilis and Chris, for joining today and your interesting contributions. And thank you all for joining today. Have a nice time. See you next time.
Bye, everyone.
Bye.